of PDT here in Brazil. And he also is going to speak today about optical diagnostic in use in oncology in other areas as well. Okay, Vanderlei?
Okay, I did not give myself this title. This was all set by Claudio. So I suppose to provide you an overview of how can we use photons to make diagnostics. Of course, we all use photons to do diagnostics. This is, you know, NMR, X-ray, they are all photons of different energies. And, but in any case, recently, we have changed the range of wavelengths and being able to do a good job in detecting diseases, detecting different metabolic levels of tissues. And this is more or less what I'm going to show to you. So my idea is not to present in detail some of the research we are carrying on. Later, I'm going to try to put you in perspective what's going on. And of course, the things I have mostly is the material I do in my own lab. So I'm going to try to base myself in some of the material we do. But you can find plenty of literature in this subject. You know, because visible photons, they are easy to handle. And today they have very good sources. You know, lasers are wonderful sources of light that you can't have in X-ray, unfortunately. Because when you have a laser, the light has some properties that reveals more than the interaction with the tissue. You know, you have the coherence, you have many new effects that you can use in your favor using laser instead of normal light. So I'm going to try to give you, this is not supposed to be a very formal. So I'm going to kind of giving a talk as a talk in the bar, trying to show you some of the main features involved in this. But I would like to finish with some proposition, because we are deeply involved not only with diagnosis, but with treatment. And in the same way you use photons to treat, in the gamma range and in the X-ray range, we also use for the treatment photons in the visible range. And this is called photodynamic therapy, and you're going to hear plenty of this in the next talk given by Ron Allison, which is not only a very good support here in Brazil, but it's also one of the world pioneers. And he comes from a group that is being the big push of photodynamic therapy around the world. So, and well, I always start showing people, and I could not, you're going to see that I put together this talk quite fast, and I can't put all the people working in my center. It's not my center, it's the center of University of São Paulo in São Carlos. We have about 7 people working in research. And this is some of them working different aspects of some of the things I'm going to show to you. And we do have, as a center belonging to the University of São Paulo, we have a small standard. And we are doing a lot of development of new instrumentations. And they are there, and we brought so we can kind of generate relation with groups in Brazil and outside that are interested in this. If we have to say one phrase to characterize optical diagnosis, I think what we're doing here is exploring the effects of cells and tissues on light. Normally, PDT is the opposite. You want to understand the effect of light on tissues. Here, we have to look to the other side of the coin. We have to look how the tissue can modify the properties of light. And how can we make a correlation of the type of tissue with the modification that was made in the light? If you are able to do that, we can identify what is in the tissue. And of course, there are many ways of doing this. All the properties of light interacting with matter somehow can reveal different and important features that can be used to identify what is there. Of course, we have to learn how to read this, and this is the main problem. But everything is on the cell. So the structure of the cell, the biochemistry going on in the cell, all this interacts different with light. That's why tissues have different colors, because they interact different with light. Right? And nobody has doubt to identify the color of a tongue and the color of the skin, because you know, it's so obvious. Huh? The biochemistry going on there, the structure is so different that helps a lot to identify. But we want to go further deep in this process and try to identify what's going on inside the cell and how the cell's arrangement can also be identified optically and so on. So this is optical diagnosis. It's used not only for cancer. Of course, cancer is our main target, but optical diagnosis is being used for many, many things. Right? Today you go to the hospital and to know how many sugar, what's the concentration of sugar or oxygen in your body, they don't need to take the blood anymore. They can just put a little light close to your skin and identify that. This is optical diagnosis. It's part of my talk. You know, everything you use optics in the sense of visible light to identify something is in my talk. Of course, I can't cover all the application because it's too many things. But there is one simple principle involved with this, and I want to show you because I know that we are not all physicists or chemists, but it's very simple to understand. First of all, light, we can't look light as some energy from the cosmos. It's impossible. We have to look light as a tool, as made of electricity, as made of electromagnetic fields, and those fields interact with matter because matter are charges. As we all learn in elementary school, before years ago, we learned about atoms and molecules in high school and faculty. Now you speak with a kid, he can tell you exactly what's the difference between carbon and silicon and water and everything. So atoms, we are in the age of atoms. Biology is atoms today. It's not a classification of classes of animals anymore. It's called molecular biology. This is what's going on. And so, you know, it's charge. Electricity and charge, they combine quite well. One likes the other. And what's happening is when light interacts with matter, it just shakes the charges. And as an antenna, these irradiate back energy. And whatever is there as matter, as atoms, as molecules, is the response of light coming out. This is the principle. Okay? When you do X-ray, you just absorb, and by the contrast of absorption, you try to identify structures. Here we can do much better because depending what's the light coming out, how is the light coming out, we can identify what is there, how much, in what way, in what composition, and what structure. But everything is based in the old good principle that electricity and charges, they like each other. And when you shake charges, they irradiate like antenna of a radio or your cellular phone. Okay? And this is the whole idea. And what you want to do is take the light that comes out, because it's very hard to get the light that goes in. The light that goes in is the light that we call absorption. That one, we can do very little with it. You doing the X-ray, and I'm going to show you that by time of flight of photons in tissues, you can also do a lot of things. But here we detect the light that's coming out. Some of the light comes out like a reflection in a mirror. Some of the light comes out passing through the tissue. And that light carries information because that light necessarily interacts with the whatever is there and gives the information. Okay? And there are many ways of light interacting with tissues, with cells, and I just put this so I can kind of give you an overview of all the features happening during the light-tissue interaction. And then I'm going to show to you that each of those can be used for diagnosis. So the first thing is, when you have a source of light, you shoot light in the tissue. Some of the light comes out. So sometimes my skin, especially in this top here, looks very shiny, like a mirror. This is what we call reflection. And this light actually interacts very superficial. And it comes out. It has a lot of information too. Okay? And then there is some light that penetrates. And then this light that penetrates finds different paths, because the tissue is made of interfaces, is made of small solid particles, is made of molecules. And that light started to go to a real trip inside the tissue. And we call this diffusion. Some of the light during this process are absorbed and are converted in different types of energy, because you are shaking those charges, you are heating up the tissue. That's why you stay in the sun and you warm up, because a part of the light is being absorbed, shaking your molecules, like samba of molecules, and this is heat. So you warm up. But part of the light goes around in a very, very complex path and comes out. This is called diffusion reflectance, or we normally call scattering, and light comes out. So sometimes I illuminate one spot and some lights can come out of the other one. This is the light that finds a very strange way and find a way out. So you have the absorption, and sometimes you have absorption followed by emission. And this emission normally changes the color of light. And this change depending on whatever is there. Whatever is there. If you have there chemicals or too many chemicals and too many different types of chemicals. And that is called the fluorescence. But you also have another one that's called Raman scattering. And this Raman, I'm going to explain to you in a few seconds, is also an important tool for diagnosis, because all this process depends on what is there and how it's arranged. So you can use it to identify the tissue. So absorption is a way of converting light in something else. Normally heat, because you absorb the light. And when you shine light in anything, you don't absorb the light. Always. It depends. Like if I shine a white flash of light in that fellow there, Didi, and he has a blue shirt. So he absorbs all the light and re-emits the blue. All the rest of the light he absorbs will be converted in some type of energy. Right? And normally is heat. And of course, if I send light and gets, I can identify that, oh, that t-shirt is blue, because he's sending back blue without looking, right? If he was inside a black box, I could do the same. So absorption is important, and depending on the composition of whatever you're dealing with, different colors absorbs. So that's why we are not transparent, not through the visible light. We are more or less transparent to very high energy photons, like X-rays, but not for visible, because we have so many molecules that always some of them are absorbing some type of light, some frequencies or some wavelengths. So absorption is important, and we can identify a lot of things like the oxygen content on the body. If the body is oxidized or reduced, the absorption of red light is different. So I can, with a very good precision, identify the amount of oxygen just by touching light on the finger, right? The same with different colors with the glycoses and so on. And now fat absorbs light in a different way. So today people are already using optical device to measure the amount of fat you can have in your tissue, right? Okay, so absorption normally is very important. And due to the absorption, I just put this because we use light to cut steel. And the reason is light is absorbed and converting energy and vaporize that target. And that's why laser is a very nice surgical tool too. Because light is so much absorbed and vaporized, you make a precise cutting and so on. So absorption normally depends whatever is there. And if you know what wavelength to use, you can identify a substance. And this absorption spectroscopy is a technique that's been around for 100 years or more. In fact, that's how they identify what's in the atmosphere. Just look at the sunlight and see what's missing in the sunlight. You identify, oh, it's missing ozone, or, okay? So this is something that's well known. Now, scattering. This is not absorption. Scattering is the deviation of light. The same way that we construct lens, because we use the refraction of light. You can imagine the tissue, light bounces or being refracted in different parts of the tissue and producing a PET that's very, very chaotic. And if I ask to you, look, if I shine light in one side of the tissue and some light come out, because I'm illuminating, like suppose I put this in my finger here. You see that my finger gets a little, and I ask to you, where those photons that are coming out, what was the PET they did? And it's a very complex PET, okay? And during this PET, you can imagine if I have a tumor here, things will be different. So don't think that scattering light is bad. No, no, it's good. And I'm going to show to you that we can use it to identify how light distributes on the tissue, because it will depend on that. And how long it takes for the light to come. You know, recently I saw a paper presented by a Russian group that they send a few laser pulses in this side of the brain and collecting different points. They identify a tumor inside. And of course, this is in a very early stage. But sooner, we're going to have full optical devices for complete body diagnosis. Okay, and with very high precision, because here we can change so many things and we can identify together scattering, fluorescence, and everything. So this is exactly what I did in my finger. If you put a very point light sources in here, light comes out in a different way, even inside the mouth. You see light is going there. And this is mainly the scattering property of the tissue. And depends on the properties of the tissue, okay? Scattering here, or in a bone, or in my nail, is all different. And depends on the color too. If I had a blue laser, the scattering will be completely different, because normally combines with absorption and everything. It's wavelength dependent. Okay, and of course, this is what I was telling you. We can use just the optical fibers and a device, and by shining light in some points, which is called S here as source, and detecting spots in different ways, you can either use the symmetry of the body, or you don't need to use the symmetry of the body. Today they have Monte Carlo kind of programs that can come and map what is the optical distribution property inside. So this is the identification of a tumor in the brain, just by scattering of light, nothing else. Okay? And of course, that's involved a lot of mathematics and physics and chemistry that you don't want to hear, right? But it involves a lot of things, including the time that takes. You see time here. And this is done by pulses. I pulse the light here, and I do what they call a time of flight of the photons, of the light portions. And by doing this analysis, I can just make this draw here that you see. And I believe that this time of flight, recently, Brian Wilson, which is a very nice scientist from Canada, came to São Carlos, and I took this from him. And this is a new thing that's called time of flight. It has a lot of potential for identification of not only tumors, but a lot of different things, like stress of the muscles, or the density of vessels that you have in one region. So you can identify a lot of things just by send a pulse of light and detect how long it takes for the pulse to come out in another point. And this is our optical diagnosis technique. Okay. Of course, there are complications, and you have to understand quite well how much of the light is lost in there. And but time of flight is interesting because you don't care about the amount, you care about the path. You always know you have some absorption, but you only care about scattering. And this is interesting. Okay. But you have both together when you're doing this kind of thing. So, and how can we use scattering of light for some of the things? Today we have a problem in doing photodynamic therapy. Because if you have a tumor, we have to guarantee that we are illuminating the tumor as a whole. And we have to guarantee that any portion of the tumor is receiving the right amount of light. So the way we're doing this is the part of the thesis of Lily, which is absent. She's working in the secretary. If you shine on one spot of light and collect the scattered light, you can come and map quite well the distribution of light. And you need that, you know. As you're going to see from Ron, PDT works. The principle is so simple. We can't question so much. But we're not doing complete right yet. We have to optimize. And I think this kind of combination of this and some of the other things I'm going to show you is important. So I believe that one of the most important use of the scattering light for PDT is to map what is the light distribution. Because if I put a lot of fibers in here, I just collect and I see the intensity as a function of position. And I can draw how is the light distributed in terms of intensity and position. Well, the next one is Raman. What is Raman? Well, if you take one single atom, the light is absorbed by the electrons, and you can't do very much. But if you have a molecule, the electron absorbs light, but as you know, the electrons in a molecule makes the several atoms to stick together, and there is a charge distribution, which we call chemical bond, that keep all those atoms together. So sometimes I shake the electrons, and they transfer a little energy to the shaking of the whole molecules in a way that we call vibration or rotation. Okay? So I send one light, one color, which is completely determined, one color completely determines the energy of the light. And then because energy goes in a different forms on the internal degrees of freedom of the molecule, the light that comes out is displaced in energy. And if I look at that light coming out, displaced in energy, I know exactly how the light was distributed internally in the molecule. And this is called Raman effect. Raman was discovered by a very important guy, Raman, and he's from India, and he discovered the Raman spectrum looking and explained the blue of the sky and the blue of the sea in a trip. Okay? And of course, you see when you send one frequency and you can see light coming out in a higher frequency, which means the molecule was already shaking. When the light interacts, that energy of shaking was transferred to the light. This is what we call the Stokes, or well, the anti-Stokes, sorry. Oh, this is wrong here, I'm sorry. This is things of the end of the night. So one of the photons come with high energy, the other one comes with lower energy. So you either giving energy to the light, to the internal vibrations or rotations, or you are taking energy from those effects of the molecule and putting the light. And this is unique related to the molecules. So if you do the Raman spectrum, you can identify what is there and in what quantity. And this is a very good bioanalysis of the tissue, because depending on the tissue and the conditions, you have different biochemistry going on. And by Raman spectrum, you can identify what's there. And people use the Raman spectrum for a lot of things, including here in Brazil, at the Univap. They are doing a very nice work, identify not only tumors, but also the clogging of vessels, and I think Christina can say something for us if you are interested, right, Christina? So it's a nice technique, but has to be done with lasers, because the laser has this precise energy and has all the ingredients necessary to deliver or to take energy from those internal degrees of freedom from molecules. And of course, is an important technique. We use that in the lab to identify a lot of things, and people now are identifying biological tissues. And more than that, variations of biological tissues. And now comes fluorescence. What is fluorescence? Well, everybody knows what is fluorescence nowadays. I send light to a substance, and depending on the structure of the substance, I send blue light, and it can give me back red light, yellow light, green light. Because if you look, I give energy to the electrons, and sometimes they first transfer some little energy to the molecule and then decay, give lights back. So if I send blue light, I can get a lot of light with less energy than the blue. And that's called fluorescence. And that's, again, very dependent on whatever is there. If it is very dependent to whatever is there, we can use to identify. And when you send light to the mucosas, and I think Christina is going to speak a lot about this in her talk on identification of mouth tumors. No, the light penetrates a little bit, and you have some light fluorescence coming out from the mucosas, some coming out from the submucosas, and so on. And if you are smart enough, you know that the tumor has modified this structure and the composition. And you can not only identify by fluorescence that you have a tumor there, but what is the extension? If it is only in the mucosas or if it's going deeper and deeper. Of course, we have a problem here is light has to penetrate, interact with the tissue, emits photon as fluorescence, and some of this photon is lost. So all this has to be taken into account. Okay. And of course, when you're doing fluorescence, you want to try to identify what is there. And this is the thesis of Didi, the guy of a blue shirt. And the idea is, it's not so easy. You have to work very hard because all this process goes on with the light penetrating, but goes on also with the light coming out. Some of the fluorescence is absorbed, some of the fluorescence is diffused, some of the fluorescence is everything. So, identification of this is a very hot topic, and I don't think we have the solution yet. But sometimes you don't care about what's there, because you know that one type of tissue has a special pattern of fluorescence. And you just identify the pattern, even though you don't know exactly what is there. But we would like to know what's there as well, because we can have more information than just the pattern. Okay. So, and then, you know, with fluorescence, it's like looking, right? Sometimes the doctor looks and sees some different of color in the skin. He knows that something is going on there. We are just looking with the different eyes in here. We're looking with a spectrometer that can identify the light a little better than the eyes of the doctor. Okay, so let me show you some of the things about fluorescence to monitor metabolic variations. Because cancer is important, but sometimes a pre-cancer stage is important too, or any other kind of lesion or disease going on, and you want to identify using fluorescence. So this is a very active field. And normally when I shine light on the tissue, some of the light in the same wavelength that here is, for instance, this will be 440 nanometers. This is blue light. Comes out. This is what we call retro scattering. It's the light that comes back. I'm sorry, it's in Portuguese, but retro is retro in any language. And then you have a big peak of fluorescence. This is the typical spectrum we get when we do fluorescence, and I think Christina is going to elaborate more on this. And if you have a tumor, for instance, or a normal tissue, this spectrum changes completely. So this is a change of pattern. And what we do as physicists, we want to understand what are the modifications. This is very radical modification, but sometimes it's not so radical. Though we want to do a mathematical processing of this spectrum. And normally what we do, we compare, you giving one spectrum of fluorescence, we compare different wavelengths, the relative intensity of different wavelengths. And by making correlation, we know that there a disease or specific disease. So basically what we do in these stages is identify patterns. We can't tell exactly the contribution. We have an idea what's contributing, but we are mostly identify patterns. And of course, cancer diagnosis is one of the most important because we are not in this stage yet, but we're going to be in a stage where we identify a few cells in the tissue. Right now, we just look in a very macroscopic way and making average, but the single cell spectroscopy in vivo is coming very soon because now we have cameras, micro cameras, with magnification of 250 times already. So, you know, we're not going to be able to identify fluorescence in a single cell. We're also going to identify where in the cell very soon, in vivo. So Christina is going to elaborate on fluorescence, and this is Christina searching for all the cancer spots in a guy that's being treated by photodynamic therapy in a situation that, you know, if you're going to collect normal biopsies here, the doctor is going to spend the whole day collecting. And the whole idea is after the treatment, how is the tissue coming back? If it is health tissue, if it is things like this. And optical biopsy is very good for that. And this is, again, an example is detection of hepatocarcinoma. And I think Ajit, a medical doctor from the medical school in Ribeirão Preto, want to explain some of this. And this is a part that we have a world patent on this. It's legal medicine. You know, because when the tissue stops living, it starts to decompose or to modify. And fluorescence is giving all that information. And this is time independent. So if you find a body, like in Rio, it's not very, we don't find bodies very often in Rio, but if you find a body and you don't know when was the moment, you can search the fluorescence in different parts, internal parts and external parts. And by converging some diagrams, you can precisely tell, with some imprecision, of course, the moment of death. And I think this will be a good revolution. We did some experiment with rats. We got a patent, and now we want to do with porks, and finally with human beings. But I believe that this is because the changes are so clear that you can go back and make the history of and find the time. Well, this is just for you to show where this is going. This is identification of soil and plants. We use fluorescence to identify diseases in plant. The amount of hydro stress, because here in Brazil sometimes it rains too much, sometimes it's dry like crazy. And in some parts of the country, some plants doesn't go. And you want to follow that. And normally you take fluorescence spectrum, and now somebody in atomic physics suggests that we should do what we call the spectrum correlation function. We take the fluorescence in one point, we correlate with the next one, and we make this integral. And this is the spectral correlation function for different diseases in plants. We didn't apply to cancer or anything. And it's clear that you have a good identification. I think one of the students, Emily, is going to present a work on that. And I believe this spectral correlation, which is very sensitive to the noise, the idea is what one part of the spectrum has to do with the other. If they are correlated, it's also a lot of information. So there is a lot of information in the noise. of those spectrum, as you can clearly see here. Well, of course, Carey and I don't know if Ajit is going to show this. This is a plot that looking the spectrum of the liver after hepatectomy. You cut part of it, it is recovering, and you are analyzing by fluorescence in the black line, and the red spots are the recovery of mass of the liver. And it's very interesting that you see in the fluorescence some waves. And these waves is well known from histological exams as the waves of regeneration in tissue. So the precision of fluorescence or optical diagnosis is very high. You can see things that sometimes is hard to see in the visual examination of the tissue. This is another work of a student. Is here in Brazil we take a lot of sun. Can we use the touch of a fiber to identify if the skin is being permanently changed? I'm not saying that's cancer yet, but maybe it's going to. So we have producing animals UV lesions, and we can identify, once you did the UV exposition, you can identify by fluorescence the evolution of the lesion in skin and the recover. And as you can see, the recover after this time is not yet on the level that start. Maybe will never be. And if it is never there back, means that you have a permanent modification. And maybe we can associate this level with a dangerous level of burning. So for countries like us that people like to get sunburn, maybe it's nice to know the level of permanent modification of the skin they are getting. And of course, following the drugs that we inject, this is called drug kinetics, is also because, I mean, it's clear identification of the chemistry going on, so you can identify quite well. Oh, I think you're going to say this is the work of Ajit, which is evaluation of organs for transplantation. You take one organ, you follow it, and you know if some big change has happened in this process. Because sometimes they take one from the donor and they implant and does turn on, right? Why did not turn on? Something modified between. And today they do a visual exam or sometimes take a little piece, but by fluorescence, I believe. And he's going to show to you that fluorescence reveals the level of mitochondrial respiration in the tissue. And you can more or less study how is the tissue degrading. Okay. Well, you're going to show this. I don't want to take your show out. So. And also you're going to speak about the steatosis, evaluation of the quantity of fat on the liver by fluorescence. And it's very, very, very clear correlation and data that you can identify. But of course, it's not only... Quanto tempo aí, Cláudio? Você não está mais? Eu estou marcando. When you do image, fluorescence is also important in image. Visual, you just send light, put filters, and collect the different colors, and then you identify regions of lesion, cancer, and any other disease you want. And this is already being well applied. And this is the cancer a spot in a bladder. This is the big tumor, and of course, by normal cystoscopy, you cannot identify those small spots, but by fluorescence, it's like seeing a little point. It's very hard to see variation. When you have a lot of light, it's very hard to see small variation. But when you have dark, seeing a few photons coming from nothing is perfect. Your eye can identify almost 1 photon per second in the dark. Now, if you want to identify a few photons missing in a trillion, it's hard. So fluorescence is good because of that. You create dark background and you collect a few photons coming out. Okay, I'm going to have to go fast on this. It's too many things. Well, this is something that we recently have done. Is if you are a biologist or medical doctor, you love to see things in the microscope. And recently, we have produced a new technique that's called chromatic contrast by continuous variation of intensity. But it's not the intensity of light, and it's not flipping some filters. It's continuously exploring the color composition, continuously. You put red light, green, and blue. This is the same device. And you shine the tissue, change continuous the composition. So this is an example of what you see in white light. And if you flip a filter in there, of course, this is a technique already used. You put a blue filter and then sometimes you put a red filter. But now you can do much better. You can explore continuous the variation of light. There is a diagram that's called RGB diagram. And if you can continuously vary the intensity of red, blue, and green, you can map the whole universe of RGB. And of course, you can reveal things that you couldn't see by white light. So this is also photodiagnostic, because you're using the birefringence, the absorption, and everything caused by light. Now in the last 2 minutes, I'm going to tell the most important thing is, can we use photofluorescence, for instance, to know the photodynamic action? So can we come and predict using photodynamic therapy how much or what the extension of the killing we're going to do? And the response, I believe, is yes. When we're doing the photodynamic therapy, we put drugs. And this drug is excited by light. And then reacts with oxygen. And reacting with oxygen produces the singlet oxygen and kills the cell. Now, the same oxygen I produce comes back and reacts with the photosensitizer, the drug I put in. So by monitoring the fluorescence of the drug I inject, I more or less can know how much photoreaction I create. Okay? This is not everything. Now, I'm going to go fast now, sorry. So each of those drugs, they have a tolerance of light, so they degrade with different amount of light you put in. So if I have one tissue with a drug and I shine light, the same way that it is reacting is also degrading. And some people use that as a dosimetry of light, because there is a relation between the amount of light and the degradation. But recently they discovered that the degradation has a good component due to the singlet oxygen we produce. So the fluorescence also is an indication of the singlet that we produce. And depending on the quantity of singlet oxygen we produce, is the damage on the tissue we're going to cause. So if we know the distribution of light, and if we measure the degradation as a function of time, we can come and tell exactly what is the extension of killing we're going to do. To do that, we need the concept of this threshold, and we have a lot of experiments that shows that we are right. And the whole idea, I'm sorry, I'm going to jump all of this, is that we inject some drug. By fluorescence, we evaluate during the treatment how much drug is still there, not decomposed by the photodynamic reaction. And that is an indication of the amount of photodynamic reaction we produce. If we know the distribution of light, we know that for each quantity of light, what is the amount of oxygen we produce in each part of the tissue. And we know exactly if that is above or below the quantity we need for the necrosis to take place. And this is what we're doing now in São Carlos, is trying to prove this concept. And I'm sorry that I was so excited by the rest of it that I compromise the most recent work we have done. But I want to stop here. And I know I speak very badly, and so I always bring a present to give to the audience. It's only one present. So this is a Brazilian cachaça. It's a very special cachaça. Small bottle, right? They don't produce very good cachaça in Big Town. And this is, that was produced for the celebration of a very important company we are collaborating. And of course, I want to offer to somebody. So I'm going to open to questions. And then if you make a question, at the end, I make a lottery and give you this cachaça. It's really good cachaça. Okay. I'm going to open and show you. It's really good. Unfortunately, I would like to bring many to give to you. So it's even good that, you know, Americans love to put in the pocket and they can drink some. So it's open for questions, and if you do a good question, you take the cachaça.
You got a question for a couple time for a couple questions, I guess. Any takers?
He wants the cachaça.
What kind of mistake can different composition of the cell cause in this kind of diagnostic?
What kind of mistake?
I mean, fazer em português, tá?
No, there are, of course, limitations. Sometimes we think we're seeing something that we are not.
I mean, if the cell has more iron or lead or
Oh,
things like
okay.
that.
Well, if you know how to excite, what wavelength to excite, and how to collect, you can identify whatever is there. You know, this is fluorescence techniques in chemical labs is used for a long time, and they identify pollutants. Like today, you know, they can monitor the whole city of Rio de Janeiro with 2 lasers scanning the sky and seeing the fluorescence, and whatever they cross and they detect, they know who is emitting pollutants and everything. So you can identify quite well. I don't know if we know how to quantify yet. That's a problem, because light that's emitted inside the cell, you know, undergoes so many different things. Now, there is one problem which I could not elaborate with the imaging when you're doing fluorescence for determining the extension of the tumor. As you know, there is one part which is clear. I don't need anything to identify, it's clear there. And of course, you do fluorescence and you identify that that's a tumor. And then there is a margin. So there is a study by some people from Sweden, and then they did the following study. I identified the tumor, and as you're going to see with Christina, almost 100%, the optical diagnostic coincide with the histological diagnostic. Almost, right? It's 95. It's okay. I call this about 100. So you have this. It's quite good. 95, 96. Now you start to go further from the main tumor in the region that you call margin. So they say that if you go some amount, the coincidence of the histological and the fluorescence goes down to 50. If I go a little further, the coincidence goes to 27. And that's a problem, because I don't want to detect a tumor and then detect a spot and say no if it is yes. And people has found in the margin, observe, far from the tumor, only 20%, 27% coincidence between the histology exam and the optical diagnosis. And the way of improving this, because there you have only islands of cells that on the microscope you identify, but the optically you're not seeing. And I think the solution for this is what I call a microscopic fluorescence spot. You have a little camera and you are just looking, counting photons coming from different spots. And I think this will be the next generation of possibilities to improve and to have really something that can be safely used in practice.
We have a winner. We gotta stop.
You have what? Você tem uma lady there.
No,
Christina.
you run out of time.
Okay, sorry, Christina. I can.